Hey guys, how you doing? So welcome to chapter 15. In this chapter, we are going to look at genotypes and phenotypes. We're going to look at how genetics result in the traits being expressed in an individual. Okay, so I'm going to break this video up into a few different parts, but the first part we're going to take a look at alleles and the dominant nature of alleles. Okay, so let's get into it. All right, genotypes and phenotypes. Now here's the outline. You can probably have a look at it yourself. We're probably going to go through that together in class anyway. Let me recap on the word alleles because it's important for us to understand what an allele is and how it affects an individual gene. Okay, alleles are variations of genes. Okay, and this whole idea of dominant and recessive relationship is based on the fact that every individual has two alleles of every gene. Yeah, so in your body, you know, your eye color, your skin color, or whatever is all determined by multiple alleles. Not just one copy of the gene, but multiple copies of the gene. Two, in fact, because you receive one from your mum and one from your dad. Yeah, the chromosomes in your body come in pairs, and one copy of it comes from mum, one copy comes from dad, which means the genes in their locations have slightly different coding instructions. Right. So if we look at flower color. This particular chromosome of the homologous pair might be for purple flowers. This one might be for white flowers. Okay, and so what that does, what that means is that the flowers now have to determine sort of which one gets expressed into the trait. Does it do purple flowers? Does it do white flowers? Does it do a combination of both? And that particular interaction between the genes is what inheritance is really all about. Okay, the same thing can be said for eye color. So some individuals might have brown eyes, some individuals might have blue eyes, and the relationship between an individual who has both the genes for brown eyes and for blue eyes needs to determine what type of relationship they have. Okay, so in that particular case, we have a word for that, and it's called heterozygous, which we'll talk about more later. And then we also have a word for the ones that have the same gene, such as all brown or all blue, and that is homozygous. Okay. Now let's look at dominance and recessive traits in individuals. Not all of the alleles are being expressed. Yeah, so you might have two copies of eye color. One that says, you know, create blue pigmentation in the iris, or one says create brown pigmentation in the iris, but not both of them will be expressed at the same time. Okay, and so there is a type of hierarchy or relationship, and that's what the dominance relationship is. It's that hierarchy of which allele gets priority is being expressed into the trait. Right? If it is a dominant allele, it means it's always going to be expressed. Yeah? If that gene is present and that gene is a dominant gene, or the allele is a dominant allele, it will always be expressed in an individual. However, it's recessive. It will only be expressed if the dominant allele is absent. Yeah, so when the dominant allele is not there, that is when the recessive allele will then become expressed. Yeah. Now, depending on the type of gene, each gene will have a different dominance relationship to its within its alleles. Okay, and there are three particular types of dominances. Okay, the first one is called complete dominance, where the dominant allele will completely mask the recessive allele, which means only one of those genes are expressed and the other one isn't expressed at all. Yeah, the dominant allele completely masks it, and the recessive allele doesn't get expressed at all unless the dominant allele is absent. There is an incomplete dominance, which means the dominant allele doesn't completely mask it, and it produces this kind of intermediate expression. You're going to kind of get half of the dominant allele trait, and you also get half of the recessive allele trait, and we call that incomplete dominance or partial dominance. They mean the same thing. Okay, and the third one is codominance. If both the alleles are going to be expressed in full, then it means that they are codominant, yeah, which we'll look at cases for each one. Yeah, let's start with understanding how we recognize alleles and what they mean. Yeah, the symbols are really important. So we're going to go back to Mendel's P's from chapter 13, and we're going to look at some of these traits. So here you can see that Mendel was able to isolate which trait was going to be a dominant trait and which one is going to be a recessive trait, right? And so flower color was also shown to be dominant or recessive. If it was purple, it was dominant. If it was white, it was recessive. The position of the flower, if the position happens along the stem, that was called an axial flower position. If it happened right at the end of the stem, that was called a terminal flower position, right? And so axial was dominant, yellow seed color, round seed shape, and inflated pod shape, and green pod colors were all dominant features, whereas the other counterparts were the recessive ones. Okay, now in letters, we use a capital letter for the dominant trait and the lowercase one for the recessive trait. Yeah, so P capital will be for purple. Use the same letter for that trait. So lowercase p would be white flower, even though you might assume that, why wouldn't they use the letter W? No, because P in this case represents represents flower color. Okay, A might represent flower position, capital A for axial, lowercase a for terminal. Now these are assigned by people. You can make this up as you go. Okay, yellow, which is capital Y, and lowercase y for green. Okay, if you use a different letter, you are now talking about a different trait. So if you're talking about the same trait, seed color, flower position, or flower color, you have to use the same letter. It's just whether it's in capital or in lowercase. Okay. Now let's look at the first one, complete dominance. Cystic fibrosis is our example that we're going to use here. I very briefly touched on it, but let me go through with you what cystic fibrosis is. It is a genetic disorder. Okay, it's congenital in the sense that it happens at birth. An individual with cystic fibrosis will have their lungs, pancreas, liver, kidneys, and intestines all affected by this particular disease. It happens in 1 in every 3,000 newborns, mainly of North European descent, and it is a clogging of the airways from a mucus buildup and an inflammation around the and increased secretions around different organs in the body, mainly the pancreas and the lungs. Okay, so it can result in pancreatitis and diabetes. You have 97% of men who have cystic fibrosis are also infertile because of a congenital absence of the vas deferens. The vas deferens is the tube that connects the testicles to the penis, which is what also allows men to release their sperm. So if the vas deferens is absent, the sperm is unable to be released, right? And so men become infertile when that happens, right? Now, let's look at the underlying cause. So here are the symptoms of that particular disorder. The genetic causes, the CFTR gene sits on chromosome 7, and it codes for a particular, it stands for cystic fibrosis transmembrane conductance regulator. Yeah, that's the name of the gene, and it creates a transmembrane protein. Now, that protein is really important. Okay? It regulates chloride ions in and out of the cells in areas like the lungs. Okay, and what happens is there are 2 alleles for this particular gene. The first one is perfectly normal, and the second one, the recessive allele, creates a non-functional protein. Yes, in other words, whatever the protein is being produced does not work and cannot move the chloride ions in and out of the cell as it's supposed to. So now here you've got 2 versions of the gene. One version creates a normal protein that works and it creates a normal human being, which is perfectly healthy. The second one is a recessive one, which creates a dud protein. The protein doesn't work, and that's what causes cystic fibrosis. Here's a really bad diagram. I couldn't find a much better one, but let me go through sort of the process, right? So the gene there, the CFTR gene, is here. Now, every gene contains segments of exon, which is the coding protein region, and the introns, on the non-coding regions within the gene, okay? The gene gets transcribed into a message, a photocopy mRNA, which then moves towards the ribosomes, where they then assemble the chain of amino acids. There's about 1,480 amino acids in CFTR, and then it gets folded and transported to the membrane, where it then forms the transmembrane. Now, thinking back to chapter 1, transmembrane means it spans the entire length of the plasma membrane, and it acts as an integral protein. Yeah, it regulates the movement of ions in and out, active transport of chloride ions in and out of a particular cell, right? There are a few different versions of CFTR, right? If you have a regular CFTR, you create a normal functioning protein, which then is able to move chloride ions in and out and resulting in a healthy person, okay? There can be a slight change, a substitution change in the 4th exon there, which results in a slightly dysfunctional protein. Still works, but it is not fully functional, and because it's not fully functional, you still get the congenital absence of the vas deferens, which means males are infertile. but the channel doesn't open properly, but it maintains some of its regular function. So it's a bit of an inconvenience, really, unless, of course, you're a male, in which case you have a high chance of being infertile. Now, here's the one we want to look at particularly, right? If there is a DNA change in exon number 10, and this DNA change is a deletion of that particular base, what happens is it shifts all of the codes afterwards forward, and results in a complete change in the amino acids that are being assembled within this protein, and it creates a protein that doesn't function properly, and it cannot regulate the chloride ions in and out, and that results in cystic fibrosis. Yeah. So here's two versions of the channel. Okay. Here is a normal CFTR channel that works fine. Chloride ions go in and out, and the chloride ions are actually responsible for removing the mucus lining around the lung. Yeah. If the protein doesn't work properly, it cannot move chloride ions in and out. Chloride ions get trapped within the cell and cannot escape properly, and then you get mucus buildup on the outside of the lungs. It also creates thickened secretions outside organs like the pancreas, which results in damage to those organs and cystic fibrosis in the individual. Okay. Now, this is an example of complete dominance, because if you have a big capital C, you create the normal CFTR gene, resulting in a functional protein. A lowercase c means you've got an allele for a mutated version, resulting in non-functional protein. Okay, you only need one copy of the capital C. Yeah, here is our lovely Punnett square. So you can imagine there, if you look at the genes for mom and the genes for dad, let's say in this case here, mom has a capital C, resulting in a normal CFTR, and dad has a lowercase c. Mom also has a lowercase c, and same with dad. Yeah, what that means is they've got 4 different outcomes: capital C, capital C, capital C, lowercase c, capital C, which these two here are exactly the same. The order doesn't matter. Okay, and then you've got lowercase c, lowercase c. Now, of these outcomes, right, what that means is the possibility or the possible outcomes of the kids is 1 in 4. Yeah, capital C, capital C, capital C, lowercase c, lowercase c, capital C, and lowercase c, lowercase c. Of the 4 possible outcomes, only 1 will result in cystic fibrosis. Okay, if you have capital C, you don't have cystic fibrosis. If you have capital C, lowercase c, you also don't have cystic fibrosis. If you have lowercase c, you do have cystic fibrosis. Yeah, only 1 copy of this capital C is needed for the individual to remain healthy. Right, and so out of these 4 outcomes, this one here, the 1 in 4 chance, results in cystic fibrosis. Okay, and so that is a complete dominance trait. Individuals with capital C, lowercase c are heterozygous, and they don't have cystic fibrosis. They do, however, carry a dud gene, but because they have 1 normal gene, they're okay. Right? Incomplete dominance, let's look at a particular flower called snapdragons. These flowers are called snapdragons. They're really cool. The flowers themselves come in a variety of different colors, but the seeds themselves are wicked. Like, if you actually look at it, they look like skulls. Yeah, they're the seeds of the snapdragons, and they look pretty freaky if you look at it close up, right? But the flower color is what we're going to be interested in. Okay, flower color is determined by 2 alleles. Yeah, if you have capital R, you produce a red flower color. If you have a lowercase r, you will produce a white flower color. Now, here are the possible outcomes. If we do the mixes, right, flowers which have 2 capital R's will produce the red flowers, and flowers which have 2 lowercase r's will produce white flowers. However, flowers with a capital R and a lowercase r will produce pink flowers. You can see there, the pink is the intermediate between the red and the white. What that means is that red flower color trait is being interrupted by the white flower trait, and it doesn't fully mask it. So some of the white is coming through in the color, and you get this kind of blend of both colors ending up in a pink, because red mixed with white results in pink. Right? And so individuals with the heterozygous trait, capital R, lowercase r, will have pink flowers. Now, this is incomplete dominance because the red doesn't completely dominate over the white. If it did completely dominate, you would expect capital R, lowercase r to produce also red colors of the same concentration as capital R, which tells you that in this case, it's not a complete dominance, it's an incomplete or a partial dominance. Yeah. And so that is a second form of dominance. Now, it really just depends on the genes, right? Snapdragon flower color would be a incomplete dominance. If you look at another one, for example, stem length might be a complete dominance. So you can see that it really depends on that particular gene whether it exhibits this particular characteristic of dominance or not. The last example we're going to go through is looking at blood type. Yeah, we're going to look at codominance because blood type is a really good example of what codominance looks like in people. Okay, I capital is the blood type. Right, so we're going to use I capital for blood type. If you have a capital I, it means you have a dominant allele. If you have a lowercase I, it means you have a recessive allele. Right? However, within blood type, there are actually two dominant alleles. Okay, you have an capital I, capital A for blood type A dominant, capital I, capital B for blood type B dominant, and you have a lowercase I for blood type O dominant. Now, how blood type works is that this gene codes for a particular marker on every single one of your blood cells, which is called an antigen. That particular marker is a glycoprotein, which allows your body to identify that as you. Yeah, it looks like a particular shape, and your body's immune system recognizes that shape and identifies that as self. Right? When you look at Year 12 biology, you understand this in a bit more detail, but that self-recognition is really important because that's how your body stops attacking itself, because when a foreign body comes in, it doesn't have that antigen marker, and your body recognizes that then as foreign, and it will attack it. Okay? So within your body, there are three possibilities. If you have a particular gene, IA, it means you have one particular antigen shape, which is determined as the A antigen. If you have a different gene, IB, it has a different shape altogether, and it's identified as IB, or blood type B. If you have O, what that actually means, it's not that you have an antigen, it means that your body has no antigen. There's no markers on its red blood cells to tell you that you have, that's you, yeah? So here are all the possibilities, yeah? Now, we're looking at multiple alleles, which means there are more than 4 possibilities. There are 9 possible possibilities, 9 possibilities that appear on our chart, okay? IA, IB, I mixed with IA, IB, I again, right? So here are the possible outcomes. If you have capital IA, you have A blood. If you have capital IA and lowercase I, you have also A blood, and vice versa, yeah? Same can be said of B, B blood, IB, capital, and BI, capital, B blood again, and 2 I's or lowercase i's resulting in O blood. If you have capital I, capital AB, what that means is you have a codominant feature, right? You have a codominant alleles, and you have both A blood and B blood, which is resulting in AB blood, right? Now, this is particularly important because what that means is that your body uses both the A and the B as a form of recognition, right? And so individuals with A blood will look out for antigen A. Individuals with B blood will look out for antigen B. Individuals with O blood, their immune system looks for no antigen. So as soon as they have any antigen on them, the immune system then says, that's not us, let's attack it, right? If you have AB, what's good is that if you have blood that is A type with only antigen A, your body recognizes that as you. If you have B antigens only, your body still recognizes that as you. If you have AB, your body recognizes that you. And even if you have O, no antigen blood, your body still thinks that's you because that's what it's looking out for, any of those markers, right? Which is why blood donation has to be so specific, right? A person with A blood can only receive other A or O blood because O has no antigen. And so your body will still recognize that as yours because there's no marker to identify that it's foreign, okay? If it's B, likewise, you can receive B blood and you can receive O blood. If you are AB, you receive A blood, you receive B blood, you receive AB blood, and you receive O blood. However, if you are O blood, you can only receive O blood because if you receive any blood that has an antigen marker on it, your body will immediately recognize that as foreign and will attack it, right? And so they say that the O blood people are the universal givers because they can give to anybody. whereas AB are universal receivers, which means they can receive from anybody, but they can only give to other ABs because their blood contains antigen marker A and antigen marker B. Okay, so that's why blood type is particularly a good example of showing codominance, right? Because that IA capital and IB capitals show a dominant feature of both antigen A and antigen B being present. Okay. A few other good examples. If you look at coat color in animals and you see splotches, that's usually a codominant trait because you're getting both one color and the other color showing up in splotches. Yeah, if you get a blend, it's incomplete dominant in terms of coat color. So that's just something worth kind of looking out for. All right, we'll stop it there. In the next lesson, we're going to look at genotypes and phenotypes. All right, catch you guys.